Alzheimer's disease is a devastating condition that affects millions worldwide, robbing individuals of their memory and independence. One of the earliest and most troubling symptoms is spatial disorientation, getting lost even in familiar places. But what exactly causes this? Recent research using mouse models has begun to unravel the neural mechanisms behind these navigation difficulties. In this video, we'll explore how tau pathology across specific brain networks is linked to spatial learning and memory, focusing on findings from the 3xTGAD mouse model. Let's dive into the science behind getting lost and what it reveals about Alzheimer's progression. To study Alzheimer's disease in a controlled way, scientists use the 3xTGAD mouse model. These mice carry three genetic mutations associated with familial Alzheimer's, leading to the development of both amyloid plaques and tau tangles, hallmarks of the disease. What makes this model especially valuable is that the distribution of these pathologies closely mirrors what's seen in humans. By observing these mice at different ages and comparing them to healthy controls, researchers can track how pathology develops and how it impacts behavior, especially spatial navigation. Spatial navigation is one of the first abilities to decline in Alzheimer's disease. Both humans and rodents with AD-like pathology show impairments in finding their way, even when using environmental cues. In the 3xTGAD mouse, these deficits can be measured using tasks that require the animal to reorient itself in space after being disoriented. This is not just a simple memory problem, it reflects a breakdown in the brain's ability to use spatial cues and coordinate activity across multiple regions, particularly the parietal cortex and hippocampus. The parietal hippocampal network is central to spatial learning and memory. The hippocampus is well known for its role in forming spatial maps, while the parietal cortex helps integrate sensory information and guide navigation. In Alzheimer's, both regions show early signs of dysfunction. By examining pathology in these areas, scientists hope to understand how their interaction, or lack thereof, leads to spatial disorientation. This network also includes the subiculum and retrosplenial cortex, forming a circuit crucial for orienting in space. Researchers studied both male and female 3xTGAD mice at different ages, 3, 6, and 12 months, alongside healthy controls. Mice were trained on a spatial reorientation task where they had to find a hidden reward using environmental cues after being disoriented. After behavioral testing, their brains were analyzed for tau and amyloid pathology using various staining techniques. This approach allowed scientists to link specific patterns of brain pathology to performance on spatial tasks, providing a detailed picture of how disease progression affects navigation. The spatial reorientation task is designed to challenge the mouse's ability to use external cues for navigation. After each trial, the mouse is disoriented and must use cues in the room to locate a hidden reward zone. The reward is only given if the mouse stays in the correct area for a set period. This task taxes the parietal hippocampal network and mimics the real-world challenges faced by people with early Alzheimer's, who often get lost even in familiar environments. After behavioral testing, the brains of the mice were carefully processed and stained to reveal the presence of tau and amyloid pathology. Researchers used specific antibodies to detect phosphorylated tau p-tau, amyloid beta using 6E10 and M78 antibodies, and other markers. By quantifying the density of these pathological markers in different brain regions, scientists could correlate the extent and pattern of pathology with the mice's performance on the spatial task. One of the most striking findings was that the pattern of tau pathology across the parietal hippocampal network strongly predicted spatial learning and memory performance. In 6-month-old female 3xTGAD mice, higher densities of p-tau-positive cells were associated with worse performance on the spatial task. This relationship was not seen with amyloid pathology, suggesting that tau accumulation in this network is a key driver of early spatial disorientation in Alzheimer's. Interestingly, the relationship between tau pathology and spatial performance varied by sex and age. Only 6-month-old female mice showed a positive correlation between tau accumulation and impairment. In contrast, 3-month-old females and both 6 and 12-month-old males showed the opposite trend. Higher tau density was linked to better performance. This suggests that the impact of tau pathology on cognition may depend on developmental stage and sex, highlighting the complexity of Alzheimer's progression. While amyloid pathology is a hallmark of Alzheimer's, this study found that its distribution and density, measured by 6E10 and M78 antibodies, did not predict spatial learning and memory performance. 12-month-old male mice had the highest amyloid burden, but this did not translate into worse spatial navigation. This finding supports the idea that tau pathology rather than amyloid is more closely linked to early cognitive deficits in Alzheimer's disease. The study also examined how different pathological markers relate to each other. Tau p-tau and amyloid 6E10 densities were generally positively correlated, especially in the parietal cortex. However, another amyloid marker M78 was negatively correlated with both p-tau and 6E10. These complex relationships suggest that different forms of pathology may interact in unique ways across brain regions, influencing disease progression and symptoms. To uncover patterns linking brain pathology to behavior, researchers used advanced statistical tools, canonical correlation analysis (CCA) and independent components analysis (ICA). CCA finds linear relationships between sets of variables, while ICA identifies independent patterns across brain regions. These methods revealed that specific profiles of tau pathology rather than single-region effects were the best predictors of spatial learning and memory performance in the mice. Within the parietal hippocampal network, the subiculum emerged as a critical region. In 6-month-old female mice, higher tau pathology in the subiculum was strongly linked to worse spatial performance. The subiculum acts as a hub relaying information from the hippocampus to the cortex and is involved in integrating spatial and navigational cues. Early tau accumulation here may disrupt these processes, leading to the observed navigation deficits. A major insight from this research is that patterns of pathology across a network of brain regions are more predictive of cognitive deficits than pathology in any single region. This supports the idea that Alzheimer's is a network disease, where disrupted communication between regions leads to symptoms like spatial disorientation. Understanding these network-level changes could help identify early biomarkers and targets for intervention. The study's findings highlight important sex differences in Alzheimer's progression. Female mice showed earlier and more pronounced spatial deficits linked to tau pathology, while male mice, even with higher amyloid burden at older ages, did not show the same impairments. These results mirror human studies, where women are at higher risk for Alzheimer's and often experience faster cognitive decline. Understanding these differences is crucial for developing tailored treatments. Tau's impact on cognition is complex. In some cases, low levels of phosphorylated tau may even support neuroplasticity and learning, especially in younger animals. However, as tau accumulates and spreads, it becomes toxic, disrupting neural circuits and impairing memory. This dual role may explain why the relationship between tau pathology and behavior changes with age and sex, and why early intervention is so important. Thioflavin S staining, which detects amyloid plaques, revealed that only 12-month-old male mice had visible plaques, and even then the levels were low. This further supports the idea that cognitive deficits in early Alzheimer's are not directly caused by plaque formation, but rather by earlier, subtler changes in tau and network function. These findings have important implications for early detection of Alzheimer's. Since spatial disorientation and network-level tau pathology appear before significant plaque formation, tasks that assess navigation and advanced imaging of tau could help identify at-risk individuals sooner. Early intervention could then target these network disruptions before irreversible damage occurs. Future studies will build on these findings by examining later stages of disease, testing interventions that target tau pathology, and translating spatial navigation tasks to human clinical settings. Advanced statistical tools like ICA and CCA will continue to play a key role in uncovering the complex relationships between brain networks and behavior in Alzheimer's disease. In summary, this research shows that tau pathology across the parietal hippocampal network is a strong predictor of spatial learning and memory deficits in the 3xTGAD mouse model, especially in females at early disease stages. Amyloid pathology, in contrast, is less predictive of early cognitive decline. These insights shift the focus toward tau and network-level changes as early drivers of Alzheimer's symptoms. Understanding how tau pathology disrupts brain networks and leads to spatial disorientation is crucial for developing better diagnostic tools and treatments for Alzheimer's disease. By focusing on early network changes and using advanced analysis methods, researchers are paving the way for interventions that could preserve memory and independence for those at risk. Continued research in both animal models and humans will help turn these insights into real-world solutions.